Hi everyone, Fritz here. In today's video, we'll be going over your negative stain. The negative stain is used for sensitive bacteria and primarily for the purposes of getting the most accurate size of a specific bacteria. And the reason why we do the negative stain when it comes to measuring a bacteria's size as accurately as possible is because this technique allows for the minimal amount of cell shrinkage or cell swelling due to its interaction with this dye. And because the bacteria that we're typically working with on a negative stain are sensitive, we will not be using the heat fixation technique. And on its surface, the negative stain may seem like the opposite of a simple stain in which we have a positively charged dye staining a negatively surfaced bacteria. Whereas the negative stain uses a negative dye to stain the background surrounding the negatively charged bacteria. So upon completion and underneath the microscope, you can expect your results to look something like this, in which the bacteria will have either a white or a clear look to them underneath the microscope, while the background will have the darker contrasting color to it. First, you will add the dye to one side of the slide. Then you'll add your bacteria in that same spot. Then you will use the feathering out technique. And the feathering out technique is to ensure that once we have these cells under observation, we have a 1-layer cell thickness. And the way that we do this is by using another slide. Imagine that we have another slide here, and this slide we will drag back almost to the edge of this slide. It will allow the dye bacterial mixture to evenly disperse to both edges of the slide. And once that happens, we will want to push the slide out. And then once you push the slide out, give it some time to dry. And this one, we just want to air dry. You will also notice as it's air drying that one side is a whole lot darker than the other side. And this is what we mean by the slide is feathered out. And looking at these two cards and imagining that this is our dye and bacteria mixture, we would want to start a little bit in front of it and then pull it back. Take a peek to make sure that the mixture has been evenly dispersed to both sides of the slide. And then once it's done that, go ahead and push it forward. You want to be gentle enough that you don't break either one of these slides, but you don't want to be too light and accidentally lift up in that process. So if you ever want to practice this technique outside of your lab, just take 2 business cards, draw a dot, start a little bit in front of that dot, like that, draw it back, wait for a second, and then push forward. You can hear the scraping of the 2 cards, and that's usually good enough. You don't want to have your card bend down. That means you're using too much leverage there. And if you don't hear that sound, like that, that means that your cards are not together. So this is a good way to practice your technique. Now, once your slide has fully air dried, you're gonna wanna look at it underneath the microscope. And you're gonna wanna look at the end that is lighter versus the side that is darker. And a good question for you to know on test day is, why is it that we look on the lighter side versus the darker side, considering that the darker side might have more bacteria for us to look at? And if you think about it like that, it might seem counterintuitive. So if you know the answer to that question, feel free to answer that down in the comment section, as well as these questions. Now, if you found this video helpful, please leave a like and subscribe. Leave any additional questions that you have for me down in the comment section below. And don't forget to check the description for additional study materials. And I'll see all of you in the next one.